Hi guys, this is the Biology Goat. So this is my first video. I hope you enjoy it. All the content that I'm going to teach in my tutorials is suitable for the AQA and OCR specifications. Most of it will also be relevant to Edexcel, but I would suggest just double checking the specification if you're doing Edexcel, just to make sure you're not learning stuff that you don't need to know. So I'm going to make some notes as I go through my explanation. I'm going to use different colours just because I think it's nice to use different colours. It helps your brain remember what the notes look like. So if you're stuck in an exam, you can kind of think, oh, what did that blue bit say? And then it just really helps to jog your memory. So what I'm going to suggest is either you make the notes with me as I go through the video today, or you can watch the video first, then make your own notes afterwards. And then maybe once you've gone through your notes a few times, start condensing those notes down onto some flashcards. So you've got like less information in front of you as you get closer and closer to the exams. So my first lesson is going to be an introduction to respiration. So I'm going to put lesson 1, an introduction to respiration. Okay, so we're going to talk about why respiration happens, like what's the aim, what's the point. So I'm going to do that first. So the aim. Okay, the aim of respiration is to release the energy that is stored in glucose or other respiratory substrate like proteins or lipids, and then transfer that energy to ATP, which is basically the fancy word for sort of like energy at A level. But basically we're going to break down glucose, a respiratory substrate, like I said, proteins, lipids, and we're going to use the energy that's released from the breaking down of that substrate and use it to make ATP. So this is what I'm going to write first. So the aim is to transfer the energy that is stored in, it's normally glucose. Most cells prefer to use glucose, and some cells, like the cells in the brain, they can only use glucose as their respiratory substrate, but it could be, for most other cells, another respiratory substrate as well, as I said. So to transfer the energy that is stored in glucose stroke other respiratory substrate to ATP. Okay, so that's the aim. The way we do this is by basically breaking down the glucose or the other respiratory substrate, like the lipid, the protein, whatever it is. And when we break it down, we're going to break bonds. And whenever you break bonds in biology, you're going to release energy. That energy is then used to make a bond between the second and third phosphate group of an ADP, and then you're going to get ATP, and then the cell can then break down the ATP as and when required, and then use the sort of like little amounts of energy released by breaking that bond in like a really useful way. But don't worry, I'm going to write all of this down for you. So basically, to do this, to transfer this energy that is in glucose to ATP, we need to do this. So to do this, the glucose, I'm just going to put in brackets, etc. So if it's like another respiratory substrate like that. So the glucose is broken down. Now, most of the time in respiration, we're going to be using aerobic respiration, and aerobic means with oxygen. So you're going to use oxygen to basically combine or break down the glucose. So sometimes instead of saying broken down, we say that the glucose is being oxidized. Oxidized in science, like chemistry and biology, also means like the loss of electrons as well, which is also a really important part of respiration. So for now, I'm just going to write, to do this, the glucose, etc. is broken down, or sometimes we say it's oxidized because we're normally using oxygen to do it. So it's oxidized, and the energy that is released. Now this is important, I need to tell you about this. I'm going to use a different colour for this. And the energy released from breaking the bonds is used to convert, so it's ADP and pi to ATP. So I just wanted to finish that bit off first. Right, first thing, this release, it's really important in biology and any other science for that matter, that when you're talking about energy, you always say it's released or transferred. You can say used, but you can't say it's made or produced or kind of lost or like destroyed, because in physics they have a saying which is that basically energy can't be created or destroyed. So you can't ever say it's made or produced, but you can release it from something and transfer it to something else. So I would definitely make sure you remember to use that word whenever you're talking about energy. And then basically what we do, we release the energy from glucose when we break the bonds. That releases this energy, and then that energy is used to make the bond between ADP and PI, and then you're going to make ATP. So why? What is the point in all of this? The point is a cell basically uses ATP as its sort of like immediate energy source, and what it will do when it needs some energy to do something, we're going to talk about later all the reasons why a cell might need some energy, or the ones that you'd need to know for A level, and that are always in the mark schemes anyway. But basically the reason we don't just take the glucose and the cell doesn't just break the glucose down and use the energy from that is if you did that, you'd release loads of energy in one go from the glucose, and actually a lot of that energy might then just get wasted or like lost as heat. And we don't want that to happen. So what we do by transferring the energy in glucose into these small molecules of ATP, it means when the ATP gets broken down by the cell, you only release energy in like kind of like small units that are like usable units, so you don't get a lot of wastage. And this is one of the reasons ATP is such a good like sort of source of energy for cells. There's quite a few other reasons as well, so I'm going to write all these down for you in a sec, okay? But just to summarize what the cell will do with the ATP, so when the cell needs energy, so when a cell needs energy, it will break down the ATP, because now the ATP is storing the energy that originally was stored in glucose. So it was stored in here, but we've used it to make ATP, so now the energy is in here. By breaking this down, now we can release that energy and the cell can use it for whatever it needs it for. So when a cell needs energy, it will break down the ATP, and it happens like this. So you've got so ATP, and so that's supposed to be an arrow. I'll do an arrow like that, and you go to ADP and PI. So it's literally the opposite of how we're making it. Now we're going to do the opposite and break it down. Now in the exam they might ask you what type of reaction this is when you're breaking ATP down. There's two things that I've seen. However, a mark scheme that I saw really recently said ignore one of them. So I'm going to tell you the one that seems to be always allowed, and that is hydrolysis. So if they ask you what type of reaction, you say hydrolysis, or you say the ATP is hydrolyzed. So I'm just going to put hydrolysis. Okay, I remember that lysis, it means to split. That's what the word lysis means. So if you're ever doing hydrolysis, you're breaking something down. We use the word hydrolysis when we're talking about other biological molecules being broken down in biology as well. So just remember, if it's hydrolysis, you're splitting something, you're breaking a bond, and then we're going to get, in this case, ADP and PI. Now, the reason we've got an I here, if you're not a chemist, is the I is for like inorganic, which just means that the phosphate group that we're going to break off doesn't have carbon in it. And in A-level biology, we just tend to write that little I after it. Just don't let it throw you. Don't worry about it too much. Just make sure you have a little I afterwards, okay? Also, just so you know, when I first learnt biology, I used to think the phosphate group was literally just a phosphate all by itself. It isn't. It's actually got some little oxygens like sticking out as well. You don't need to know the structure, but you do need to know, because of those oxygens, this actually is negatively charged, which actually means ATP is a polar molecule, and that's really useful because it means that it's soluble in water, and it's soluble in like the cytoplasm of a cell. And this is one of the other reasons why ATP is so like good at being an energy source for a cell. Okay, so what I'm going to do now is just write down the sort of like main reasons why a cell uses ATP as its main energy source rather than using something bigger like glucose. Just why doesn't it just use glucose as it is? What's the point in going through all these reactions to make ATP? So let's put here like a little sort of like heading that says, why is ATP a good energy source for cells? Okay, so a few of these things I've already mentioned, but I now want to write them down. So basically anything that I write down is the stuff you need to remember, okay? So the first reason is because I've just said this is negatively charged, which makes this a polar molecule. It means that one, ATP is small, it's small anyway, but it's also soluble in water, or like soluble in the cytoplasm of a cell, which means that it's very happy to just be there in the cytoplasm. So let's write this one down first. So ATP is polar, and that's because this has got a negative charge. Okay, that's supposed to be an explanation more like that. ATP is polar and small, so is soluble. Obviously that's really important. If it's not soluble, it's not probably going to be very useful to be inside a cell if it can't actually dissolve in like the cytoplasm or the liquids that are inside there. Okay, the next reason is the bond that is between the second and third phosphate group is really unstable, so it's really easy for a cell to kind of like cause it to be broken as and when energy is needed. So I'm going to put the bond between the second and third, if I just put PI, is unstable. So it basically breaks really easily. When energy is needed. It's not hard to break, so cell thinks I want a bit of energy, I need to, I don't know, make some new proteins. It's really easy to break this bond and get a little bit of energy for whatever you need it for, to like make a peptide bond or something like that. So that's the next thing. However, kind of coming on from here, and this is what I talked about with the glucose earlier, because you're only breaking one small bond in the huge scheme of things, the actual amount of energy that you release from this is relatively small. So you're just going to release a small amount of energy that the cell can actually use in a really useful way, and that you're basically preventing energy from being wasted. So say we just got the glucose, broke all the bonds in glucose in one go, you'd get quite a lot of energy released in one go, probably too much for the cell to use, like at that time as that energy is released. So a lot of the energy is going to get wasted, which would make the process really inefficient. So this is a much more efficient way of doing it. So I'm just going to put, however, only a small amount of energy is released, energy is not wasted. So it's kind of releasing just the amount that you need for, I don't know, to make a peptide bond, but you're not getting like so much energy that you get your bit to make your peptide bond, but then you're also wasting loads. The other issue with this, if you did waste a lot of energy, a lot of that energy is released as heat. And if you're wasting a lot of energy, if it's being released as heat and not used, for example, to make a peptide bond, it could increase the temperature of the cell. And if you start increasing the temperature of cells, you might then start doing things like denaturing enzymes, proteins, disrupting the phospholipid bilayer. The cell's not going to be very happy. So this is kind of an add-on from this as well. So also, if lots of energy was released like in one go, lots would be like wasted as heat, and this is bad because it's going to increase the temperature of the cell. So I'm just going to put could, up arrow means increase, I like to use shorthand to make my notes quicker. So could increase temp of cell equals denature. Don't forget that as well as enzymes, proteins can get denatured too. So if you get your hair, which is keratin, a protein, you put it in straightening irons for too long, you're going to denature your hair and make it go all frazzled permanently. So you can denature proteins as well as enzymes, and a lot of students forget that. So could increase temperature of cell equals denature proteins, stroke enzymes, and disrupt. If I just put, guys, biler instead of phospholipid biler to make it shorter, but I mean the phospholipid biler when I talk about that. So the next reason, why else is ATP useful? Okay, the next reason, I'm just trying to find the right colour pen so I can kind of keep this a bit consistent, is that ATP is also really easy to regenerate. So once you've broken it down into your ADP and PI, you can quickly regenerate it by joining these two back together again. Or you can do it in a way that is called substrate level phosphorylation, but that's normally very similar. You just donate a phosphate group from another substrate, it's donated to ADP and you make ATP. We'll go into a lot more detail later about the two ways in which you can make ATP and exactly how they work, but I'm just going to put ATP can be like resynthesized quickly. So once the cell's broken it down, you can quickly get some more ATP. I'm just going to put for now via, it's either ATP synthase, which is the enzyme. You'll learn all about this later on, or something called substrate level phosphorylation, which again I'll go through in more detail later on. Substrate level phosphorylation. These are generally the two ways you can make ATP. Okay, a couple other things and then we'll sort of like could do a little bit of a conclusion for this lesson. So when ATP is kind of like needed to be broken down, so the cell's got energy for whatever it needs it for, the energy can be released like really quickly and really instantaneously. The cell doesn't have to wait for ages for that energy to get released. It's just kind of there, readily available. And also, this is it's really important for you to know as well that ATP is not really there to store energy. It doesn't tend to stick around for a long time inside a cell. It's not, I mean, a cell stores energy by storing it in bigger biological molecules like glucose, like glycogen, like your triglycerides. The function of ATP is not to store energy. It's so energy can be released quickly and when it's needed. So once the ATP is being produced, by like ATP synthase, which is happening in respiration, so is substrate level phosphorylation also happening in respiration. So once it's being made, it basically is normally used quite quickly. It doesn't kind of sit around for very long, and then the energy is used and it can be released really quickly. So I'm going to write this one down. Then one more thing, then we'll do a bit of a wrap up for this session. So the next thing I want to write down is ATP is usually used quickly once it's been like produced and the energy is released like quickly or instantaneously. Which, like I said, it just means that the bond breaks, like, so you're in this sort of form here, and that energy is available immediately for the cell. It's like an instantaneous thing. The cell doesn't have to wait around for the energy to come along later. But the ATP also, because it's not really there to store energy, it generally does get used up quite quickly. So the energy can, either way, it's instantly available because the ATP is going to get broken down quickly, or because once it's broken down, the energy is there and ready kind of straight away. Okay, one more thing I want to tell you is that also sometimes this phosphate group, when it gets broken off this ADP here to make this phosphate group is sometimes used to phosphorylate. Phosphorylate just means add a phosphate group to. So you can use the phosphate group to phosphorylate other like molecules in the body, like for example enzymes, or nucleotides, and that activates often enzymes or nucleotides, so they're kind of ready for use. So it's like a kind of like extra thing that ATP is just really good for, not only so energy can be released really quickly and so it's readily available for the cell, but also because this phosphate group can then phosphorylate other molecules and activate them so they're ready to do their thing, whatever it is. So I'm just going to put the PI, so the phosphate group there, is often used, I'm going to have to squeeze this in now, to phosphorylate, and I'll just put e.g. enzymes activating them. Often when an enzyme or a protein is made, it's not often active straight away. Something has to happen first in order for that enzyme to get activated. One of the main ways of activating an enzyme or a protein is by adding a phosphate group to it or several phosphate groups to it. So the PI is often used to phosphorylate, e.g. enzymes activating them. Okay. Right, that's quite a lot of information for my first video, so I'm going to stop this one here. So just a bit of a summary of what we've talked about. So we've talked about the aim of respiration, what's the whole point. The aim is that we want to transfer the energy that is stored in glucose or something like lipids and proteins into ATP, and then when the cell needs some energy for whatever, which we're going to talk about in the next session, it will then break down the ATP. And the importance of this is, I can't emphasize this enough, this is so important, because if the cell just broke down the glucose, you'd release too much energy in one go, a lot of it would be wasted. A lot of it would be wasted as heat, the temperature of the cell is going to go up. This is bad. So by having it in ATP, it's like in small, like usable little units, so then the cell can get just what it needs, just as and when it needs it, and you're not wasting a lot of energy. So we're getting the energy. The whole point of all these reactions we're going to talk about is going to get the energy from the glucose, transfer it to ATP, and then when the cell needs the energy, it will break down the ATP rather than directly breaking down the glucose or other respiratory substrate. Guys, I hope that makes sense. I'll upload another video very shortly. And the next video, we're going to go into what a cell would need energy for, why a cell would break down the ATP, and I'm also going to go into all the things that are needed for respiration to happen, so different coenzymes and things like that. Also, if we've got time, I might quickly do the structure of mitochondria, but we'll see how we get on. Guys, I hope you enjoyed it. See you soon.